Assalamu alaikum my dear students. Hope you are fine by the grace of Almighty Allah. Welcome to NIVS Homeschool. I am Nasima Akter, Assistant Teacher of Science, National Ideal English Version School, Morning Shift. Dear students, today I am going to take a biology class for class 10 students. Dear students, our today's topic is chapter 12. Let's see today's topic from here. Students, our today's topic is chapter 12. Today we will read some topics of this chapter, and this is part 2, and today's lecture number is 6 number lecture, and SW number is also 6. So dear students, let's begin the class. Students, this is chapter 12 and the name of this chapter is Heredity in Organisms and Evolution. Heredity in organisms and evolutions. Here is written, structural and behavioral traits from both the parents, mother and father are passed to the offspring generation after generation. We will learn in this chapter how traits from both the parents are transmitted to the offspring. We will also learn about the existing classes in the living world which emerge from their ancestors through evolution. And express their visible form of present existence. Now come to here, this is page number 251. Here is heredity in organism. All the organisms in the world are characterized with their own unique features. Features of individual organisms pass to the next generation and evolve in them. This natural principle is applied to all organisms in the world. This is why rice plant grows from rice seed, mangoes form their seeds, jute does also the same way. This way, the characteristics of species are maintained in offspring through generations. Heredity is the passing of traits from father and mother to their offspring through generations. In-depth discussions and research concerning heredity are done in the special branch of biology called genetics. Now come to here, this is component carrying heredity materials, behavioral features to the offspring from generation to generation. Here is the features of parents are transferred to their offspring through hereditary materials. These are chromosome, genes, DNA and RNA. These are discussed below. Look at here, here is the figure, location of chromosome in nucleus. This is the nucleus and this one is the chromosome, and here is chromatin, this is histone and nucleosome, and DNA helix. This one is the DNA helix, double helix. Now come to here, this is page number 252, chromosome. The main heredity materials is the chromosome. It is spread in the nucleoplasm of a nucleus as a thread-like structure named the chromatin. The scientist Strasburger first discovered the chromosome in 1875. Across species, the number of diploid set of chromosomes, 2 set. Diploid means 2 set chromosome. Here, 1 set from the father and the other set from the mother. Maybe 2 to 1600 in length. A chromosome may be from 3.5 to 30.00 microns. 1 micron is equal to 1 by 100 millimicron. And in width, 0.2 to 2 microns. The function of chromosomes is to carry genes which control the characteristics of organisms to the offspring from the parents. Colors of human eyes, nature of hair, composition of skin, etc. continue intact through the follow of heredity carried by chromosomes. This is why chromosomes are designed as the physical basis of heredity. Now here is DNA. DNA The main component of chromosome is deoxyribonucleic acid DNA. Deoxyribonucleic acid DNA. It is usually a double-strand spiral structure of polynucleotides. Look at here, this is the DNA double helix spiral structure. One strand is complementary to the other. This one strand is complementary another strand. In it there are 5 carbon sugar, nitrogen based here is adenine, guanine, cytosine, thymine and inorganic phosphate. Students, look at here, I have drawn here the nitrogen based present in DNA, and they are adenine, guanine, cytosine, thymine. Look at here, adenine structure is like this. This is adenine. Shortly we can write A, capital A. And here is guanine structure, and we can use capital G for express this guanine, and they are purine base, nitrogen base. They are called purine nitrogen base, adenine and guanine. And here is cytosine. Cytosine structure is look like this one, and this is cytosine, and we can express this one capital C. And here is thymine. Thymine structure is here, and we can express thymine with capital T. And they are cytosine and thymine, they are pyrimidine. This one is purine. Purine is adenine and guanine, and pyrimidines are thymine and cytosine. And pentose sugar is here. Pentose, that means 5 carbon, they are sugar, they are called pentose sugar. This is the structure of pentose sugar. And another here will be inorganic phosphate. Students, if you look at this figure, you can see here is the pentose sugar, and here is the nitrogen base adenine, always joined with thymine with 2 hydrogen bond. Adenine joined with thymine, 2 hydrogen bond, and here is always guanine will join with cytosine with 3 hydrogen bond, and here is pentose sugar, and here is inorganic phosphate. Like this way they are joined to each other. Now come to here, this is page number 253. These 3 components collectively are called nucleotide. So if these 3 components collectively is called nucleotide. DNA is stable substance in a chromosome. In 1953, American scientist Watson and British scientist Crick first described the double helical structure of DNA, and for this contribution both of them won the Nobel Prize. Nitrogen based are 2 types, a purine and a pyrimidine. Adenine and guanine are purine, and cytosine and thymine are pyrimidine. The adenine of one strand bonds with the thymine of another strand by 2 hydrogen bond, and guanine of a helix connected with the cytosine of another helix by 3 hydrogen bond. This bond is always developed between a purine and pyrimidine. So a strand of DNA is complementary to another strand but not identical to it. A complete twist in a helix is 34 angstrom long, and in a complete twist there are 10 nucleotides. So the length between the 2 adjacent nucleotides is 3.4 angstrom. The 2 strands of polynucleotides in DNA are positioned anti-parallelly. It looks like rungs in a twisted ladder. The bases are connected flatly and horizontally in position from the main axis. The external 2 strands or 2 axes of DNA are composed of consecutive arrangements of sugar and phosphate, and internal nitrogen base are plainly aligned. In fact, DNA is a precious thread, but in a prokaryotic cell, DNA is usually circular and the surface area would be a few micron to centimeters. It is composed of thousands of nucleotides and the diameter of the twisted helical structure everywhere is 20 angstrom. DNA is the main component of and chemical carrier of heredity, and the DNA is the true structure of carrier of the behavioral characters of organisms, and it directly carries the characteristics of parents to their offspring from generation to generation. Students, I have finished my today's topic. Now I will give your HW. Students, your HW number will be 6 number, and your HW will be make 5 knowledge based question and answer. It will be your HW number 6 will be make 5 knowledge based question from this topic, based on this topic. So dear students, I hope you have understood today's topic. Keep practice it at home and do your SN HW in your HW copy.